Hello, everyone, and welcome to the CGC webinar series for 2024. I am excited to have this webinar on somatic structural variant detection via HiFi sequencing in pediatric hematologic malignancies. First, a couple reminders that the CGC 2024 annual meeting is August 4th through the 7th, and it held at the Hyatt Regency at St. Louis at the Arch. Please remember that early registration ends on May 21st. So I would like to see everyone there this year. So I hope you get to registering. Second is that another webinar in our webinar series is the cytogenetic laboratory automation, implementation, benefits, and roundtable discussion. This will be also held on May 21st. You'll see emails about it as we come closer to the date. And then lastly, I would really like to promote that the CGC has a co-sponsorship with the University of Wisconsin for the CGC Genomic Case Series. This is a monthly case report, essentially, and we really encourage fellows, residents, PhDs, anyone to present cases. These cases may be any type of genetics, constitutional or acquired. Additionally, it could also be presenting new technologies that you brought into the lab. The presentations are only 25 minutes, and I really encourage this as a great opportunity to share cases, explain concepts, and meet others in the field. This QR code will take you to a landing page where you can submit a case. Please snap it for yourself or pass it along to your fellows or trainees. We really do have a fun time at these meetings, so I do encourage that people have their fellows present. Okay, now on to why you all are here. Today's talk is somatic structural variant detection via HiFi sequencing in pediatric hematologic malignancies. Drs. Midhat Farooqi, Ayse Keskus, and Dr. Lisa Lansdon, they are presenting today. Dr. Midhat Farooqi is an associate professor of pathology at the University of Missouri, Kansas City School of Medicine. He's also the director of molecular oncology for the Department of Pathology and Laboratory Medicine at Children's Mercy, Kansas City, as well as the Genomics Medicine Center at Children's Mercy Research Institute. Dr. Ayse Keskus received her PhD from Bilkent University in Turkey. She is currently working in the Comogrove lab at the National Cancer Institute, also known as the NCI, as a postdoctoral fellow. And then finally, Dr. Lisa Lansdon is an assistant professor of pathology at the University of Missouri, Kansas City School of Medicine, and an assistant director of molecular genetics laboratory in the Department of Pathology and Laboratory Medicine at Children's Mercy, Kansas City. I'd like to remind everyone, please put all of your questions in the Q&A as we are not monitoring the chat, and we will answer all questions at the end of the presentations. And with that, I'd like to present Dr. Farooqi. Thank you.
Thank you so much for the kind introduction and the invitation. Thanks all of you for joining. Hopefully my slides are coming through okay. Perfect. So I'll be taking the first part of the talk, and I'll mostly be going into the details of HiFi sequencing. But first, disclosure. So Dr. Keskus and Dr. Lansdon have no disclosures. My disclosures are that I have research collaborations with ARIMA, 10x, and PacBio to apply those technologies to patient samples. And I have also received travel support to give talks at 10x Genomics and PacBio about those collaborations. So both Lisa and I work at Children's Mercy, Kansas City. So just a little bit of background on our hospital and what we do and how we got into HiFi sequencing. So we're a almost 400 bed exclusively children's hospitals in the middle of the country with almost 800 faculty. And we are known for one of the things is having a freestanding Center for Pediatric Genomic Medicine, now called the Genomic Medicine Center. And this was established in 2011 and then really came on the national radar with the announcement of rapid whole genome sequencing for patients in the NICU, led by Dr. Stephen Kingsmore, now at Rady. And then the Genome Center is now part of this shiny new building, which is the Children's Mercy Research Institute. And the flagship project for this research institute is Genomic Answers for Kids, led by Dr. Tomi Passinen. And this project was to basically create a large sequencing repository for pediatric rare disease of almost 30,000 children, and then also their parents and potentially also their siblings, with a goal of sequencing 100,000 rare disease genomes. So we began doing HiFi sequencing on these patients in the fall of 2020, and we've completed the most rare disease long-read genomes that I'm aware of to date, so over 1,500 of these so far. And this was published in Genetics and Medicine almost a year and a half, 2 years ago, detailing the analysis of the first 1,000 rare disease long-read genomes. And then we recently, in October of last year, also validated HiFi sequencing as a clinical test, again, for patients with constitutional rare disease. We've also started applying this to cancer samples, so that's largely what the talk will be about today, using a tool developed by the Kolmogorov lab at the MCI and Dr. Keskus, so that will be the second part of the talk. And then Dr. Lansdon will talk about our application of HiFi sequencing to the initial cohort of some leukemia samples. So what I will be talking about is the ins and outs of HiFi sequencing. And so just as a bit of a backdrop, first generation sequencing, Sanger sequencing, you are all largely familiar with, as with second generation, aka next generation sequencing. And so the topic today is third generation or next gen sequencing, using one of the two commercially available platforms as far as single molecule sequencing, and that is PacBio and Oxford Nanopore. So we'll be mostly focusing on the PacBio technology, which is also known as HiFi. And the main takeaway is the difference between third generation sequencing and second generation sequencing is that third gen sequencing is the readout of a single molecule of DNA in real time. It's also massively parallel. It also, there's a lot of similarities. It's also sort of like fluorescent detection based in the case of HiFi, but the key thing to know is it is a single molecule and real time. I'll largely be comparing to next-gen sequencing methods as a point of reference. So if you're familiar with that, you'll largely be familiar with HiFi, though the devil is in the details. So just like with NGS and the general steps of extraction and library prep and sequencing and analysis, the same holds true for third gen sequencing. And these different components largely are fairly similar. As far as extraction goes for HiFi sequencing and ONT, it is preferred to have high quality, high molecular weight DNA. And for PacBio HiFi sequencing, the standard input amount is 5 micrograms. Now, interestingly, we have tested stored samples. So, DNA that we have isolated by a range of different methods, so TKM, Promega, QIAGEN, chemogen, et cetera, et cetera. And all of them so far have worked on HiFi sequencing. So it's not that you necessarily need a totally separate isolation in the case of like OGM, for example, where you need primary cells as a source, or Hi-C sequencing from ARIMA. So we have been able to successfully use a range of stored DNA samples from a range of different tissues and sample types, including buccal, as well as bone marrow, blood, tissues from soft tissue, brain, et cetera, et cetera. So it's been a fairly robust input from a technical standpoint. You just need a decent amount of DNA to begin with. And if the quality is a little bit lower, then cleanup and maybe 7 micrograms is needed to kind of overcome those issues. However, if it's very fragmented or very poor quality, then HiFi sequencing is not the best, because we're shooting for 15 to 20 kb fragments. So that's a little note on extraction. Library prep is also similar to NGS in that you undergo a lot of the same steps. So quality control of your DNA sharing, library construction, site selection, annealing and binding of primers, adapters, etc. So this part can be automated, can also be done manually. The specifics are listed here. I won't go into too many of those details for the sake of time, but the key takeaway for library prep to know is that the goal is to create these smart bells. Smart, again, standing for single molecule real time. And these are essentially stable, mostly linear DNA molecules that in practice act as a circle. And we'll go into that in a little bit. But essentially, you will start with double-stranded DNA. Through the library prep, will create these smart bells, and then you will anneal primers and the polymerase in preparation for sequencing. So with that, this is just a comparison as to what things look like. So there's a Illumina flow cell on the left, and this is a PacBio Smart cell on the right. And then the instrumentation, NovaSeq X Plus on the left, and a Revio on the right, which is the latest instrument from PacBio. Both are sort of floor standing large instruments, about 5.75 feet high. And this is a picture of the stage. So relatively minimal on the inside. There's the 3 things that you add are sort of shown in the front, which is a sequencing plate, pipette tips, and the smart cells themselves. And then the sequencing and some of the data processing is done on the instrument itself. So the smart cell is basically a flow cell in its newest design, and this gold region, if you were to zoom in, you would see all of these tiny wells perforated into metal that are known as zero mode waveguides. And this is essentially the chamber in which the sequencing reaction is taking place. And it is known as a waveguide because it's guiding a wavelength of light. And I'm going to say zero mode is a reference to the fact that it doesn't really allow any light to pass through it. So you can illuminate it, but it does not allow for light to escape, and basically contains it in this channel, which then allows for fluorescent detection, as you'll see in a little bit. And then the detection volume of this is 20 zeptoliters, or 10 to the minus 21st liters. And so it's basically capturing a individual sequencing reaction. And so the library and the smart bell polymerase adapter primer complex that you created during library prep is basically immobilized to the bottom of this ZMW. And then the DNA molecule is read. And as the polymerase catalyzes the addition of more nucleotides to the template that the read strand that it's going to create from this template. And so fluorescently labeled nucleotides are added into this DMW, each labeled with a different colored fluorophore. And as they get incorporated into the new DNA strand, then the light pulse at the bottom of the zero mode waveguide will cause the fluorescent molecule to fluoresce, and that is cleaved off as the base is incorporated. And then that is what is read by the instrument. So these little pulses of light as the individual nucleotides get incorporated is the readout for each strand of DNA that is in each zero mode waveguide. And one smart cell contains millions of these VMWs. So the latest iteration is 25 million smart cells. So essentially 25 of a million of those smart bells, one each is being read in parallel. And so the polymerase is essentially using this smart bell as a template and creating new DNA off of it. And as you incorporate the nucleotides, you get those different kinetics that are read in real time. And that's the readout from the instrument. And that's how it sequences. Interestingly enough, there's also kinetics involved. So if there's a space in time between these different pulses, since we know the speed of the polymerase, that is what allows for detection of DNA modification. So you can also get information about methylation of nucleotides, et cetera, with this readout. And then the other key thing to know is that the quality is increased due to these circular consensus reads. So basically that polymerase is reading one smart bell over and over and over and over. So this, as it does this, the software on board basically takes this read and collapses it into one consensus read, which is known as a HiFi read. And that's where one HiFi read has a quality of Q30, and which is 99.9% accuracy. But you can also see some reads falling to slightly below that, and some reads going up to Q40 and Q50 in quality. And then the alignment and variant calling is through a pipeline that PacBio has put out with different tools for both alignment, de novo assembly, variant calling, and variant visualization and interpretation that I'll leave more to Dr. Keskus to discuss. And then in the little bit of time I have left, it was to talk about sort of the metrics of HiFi sequencing. So this is essentially one Revio instrument is able to do 1,300 genomes per year, or basically 4 genomes per run. So one Revio can do 4 smart cells. So you can get one full human genome with reads 15 to 20 kb in length at a coverage of 30x in one run. So 4 human genomes in about 24 hours. And this is the contrast with the NovaSeq X, which is about, with the largest configuration, 64 whole human genomes, also at about 30x in 2 days. So just to give you a relative idea of throughput. And then for cost, this is a real-time kind of price that you could pay at a certified core facility, but this includes sample, QC, library prep, slide selection, et cetera, et cetera. And this is an external price, so you can potentially see prices as low as $1,600 The list price for reagents and consumables is around $1,000 from PacBio. So if you add in labor and overhead and other things like that, you're probably going to be looking anywhere between $1,200 to $1,800. And then in contrast, a short read whole genome, again on the X Plus, largest configuration, sort of total cost is approximated at around $640, but this is again anywhere ranging from 400 to 800 ish to give you an idea of cost estimates. And then both are likely to fall in the upcoming future. So that was mostly my section of the talk. I'll turn it over to Dr. Keskus, which she'll focus in on some of the key benefits of doing long-read sequencing, especially for cancer, which is the detection of structural variants and long-range phasing. So with that, please take it away, Ayse.
Hello, thank you. I'll continue with the detection of structural variations using long-read sequencing. Structural variations are large alterations in genome. One important feature of structural variations are they're highly heterogeneous. The size of the structural variations can vary from 50 base pair to entire chromosome, and they can be very simple like deletions, insertions, or they can be complex structural variations defined by multiple breakpoints. Here are the two well-known examples, chromotripsis, which is a reshuffling of the entire chromosome, and chromoplexy, which is a synchronized translocation event between multiple chromosomes. Although the number of structural variations is much smaller compared to small variants in the genome, the functional impact is much higher, especially for the complex structural variations. Therefore, they do not occur very frequently in normal genome. The one mechanism leading to formation of this complex SVs is genomic instability in cancer. So this is from a relatively recent Peacock paper. They analyzed thousands of patients from different cancer types. In here, each of these small plots are from different cancer type, and each color represents a different SV type. This is a very complex figure, but I want you to focus on this gray group. They are complex, unclassified structural variations, meaning we know there is something happening, but we don't really know what. Considering the functional impact of structural variations in cancer, which can be used for diagnosis and treatment stratifications, we still don't know a lot about these structural variations. But all this analysis are done using short-read sequencing, and long-read sequencing can fill the gap and help resolve some of these complex events. You may ask how. So let's say we have this one junction between chromosome A to chromosome B. Only with this information, the reasonable inference would be it's an unbalanced translocation with a single rearranged chromosome between chromosome A and chromosome B. But what if we have two other junctions, chromosome B to C and A to C again? Now we have a completely different view. Initially, we thought it as like, unbalanced translocation, but if we incorporate information from all these other junctions, it can be a single event, a template insertion cycle, chromosome A to B, B to C, and C to A again, with a deletion in chromosome A. Or it can be a chromoplexy, which leading to 3 different rearranged chromosomes. Or it can be a reciprocal translocation event between chromosome A to C with an insertion from chromosome B. Now we have like 4 possible scenarios for these junctions. And for short-read sequencing, it is challenging to resolve such events. But with long-read sequencing, we not only identify junctions involving its event, but we can also identify the most likely structure of the rearranged region using reads spanning through multiple junctions. The impact of long-read sequencing for SVs extends far beyond the complex ones, actually. One of the most critical improvements that we get with long-read sequence is improved mappability. And which is a well-known case that for the repeat-containing regions, short-read sequences are not doing a good job. But with long-read, we can, through the reads spanning through these VNTR regions, we can fully resolve these VNTR regions. But mappability is not only limited to VNTRs, the other critical aspect of it is pseudogenes. Here we have gene A and the pseudogene A prime. And if you look at the alignments with short-read and long-read sequencing, with short-read sequencing, the alignment tools cannot distinguish reads from gene A to A prime. But with long-read sequencing, since we have a longer reads, we can map all the reads accurately. It gets even more important if we have a mutation or variations in one of them. Like in this case, we have a gene conversion between A and A prime. With short-read sequencing, again, since we cannot identify the reads accurately, the pattern that we observe with gene conversion would be very similar to a normal case. So we cannot separate between normal to mutant. But with long-read sequencing, we clearly can see these junctions from A to A prime and easily identify this event. The third feature that I want to highlight is improved phasing. On the right, this is an IGV screenshot. The top panel here is the shows the coverage, and the bottom panel shows the reads from normal samples. Each of these vertical lines are SNVs. Using the germline SNVs, we can directly phase each read, and here the yellow is haplotype 1 and purple is the haplotype 2, representing maternal or paternal haplotype. You can also clearly see the SNVs used for phasing. In here, the green SNV is specific to haplotype 2, while in here, green and blue SNVs are specific to haplotype 1. After we get this phasing, we can extend it to the tumor sample, and now we also have haplotype 1 and haplotype 2 in tumor, and you can clearly see there's a phase breakpoint of a somatic rearrangement in haplotype 2. So in theory, it all looks good and nice, but why do we need that in clinics, right? So this is actually from a clinical example. Let's say we have these two deletions, one from exon 1, and the other one is exon 2 of a very important marker gene. And when you just look at this view, it's impossible to say if these two deletions are on the same allele and we have a wild type allele or not. But if we phase these reads, now it's clear that both deletions are on the same haplotype, and we have a wild type allele, which is haplotype 2 in here. That means we're going to have a functional protein in the sample. The additional layer information that we get from phasing is all the reads from haplotype 1 is supporting the both of the deletions. That means it's possibly a clonal event which is shared across all the samples, all these cells that we use for sequencing. So with all these advantages, unfortunately, most tools for the long-read SV calling are not designed for cancer genome, so we develop Severus. I'm going to briefly describe our results and also the features, but we also have preprints. If for more details, you can check. And Severus is also available on GitHub. Severus has 3 unique features distinguishing our approach from the other long-read methods. The first one is Severus does not only work with tumor normal matching data, but it also accepts tumor-only or multi-site tumor samples. So as I described, it would be a waste not to use phasing information that we get with long-read sequencing. So we incorporated that and we provide haplotype resolved phased calls. Anyway, the last step, we also cluster and classify complex structural variations, just like I described from chromosome A, B, and C case. But before we go through all these additional features, let's check how Severus is doing compared to other tools. To evaluate the performance, we sequence 4 breast and 2 lung cancer cell lines and their corresponding normals with Illumina, HiFi, and ONT. We run 3 Illumina colors, Manta, SVAB, and GRID, and 4 long grid colors, Severus, Savannah, Cinefos2, and Anomaly SV. At the end, for each cell line, we generated 11 VCFs from 3 different technologies. Since we don't have ground truth for the cell lines, we generated confident call set as true variants are supported by at least 2 technology and 4 tools out of 11. We use Midhat to generate confident call set and then compute F1 scores for each VCF. Here is the results for H2009. Here, each color represents a different technology, and each bar is a different color. And x-axis is F1 scores. As you can see, Severus outperformed in both PacBio and ONT. Here is the rest of the results, and consistently, Severus has the highest accuracy in all cell lines. Since we have this very nice set with all 3 different technologies, we further break down the HVs into different categories to compare the performance of each technology and also each color. It's a complex figure, but I'll just walk through. So in the first one, let's focus on the repeats and the insertions. Again, color represents the technology, and each of different shapes a different cell line. And y-axis is the F1 score. Insertion, we found that like insertions and like tandem repeat segmented duplications, Illumina colors are not doing quite well, which is no surprise. But the interesting one here is this Brecan homology. In this group, both Brecans are in the same repeat element, like line 1, but in a distinct position in genome. With long-read sequencing, we also detection of structural variations also improved. And Severus has the highest accuracy for all these repeat-containing categories. For the time constraint, I'll skip the rest of it, but I want to show you the last panel, which is the basically rest of structural variations as we described as not challenging regions. Because as you can see, most of the tools have very F1 scores close to 1, except Cinefos2. And Cinefos2 is the only tool that is developed for germline calls, and it is one of the best performing tools for germline SVs, but performs very poorly for cancer genome. It is because the cancer genome has its own features and it requires a cancer-specific SV caller. But the good news is Severus outperforms in all these colors, although I didn't show you, please take my word for it. So since now we can detect the structural variations accurately, now we can move to the complex SV clustering because cancer genome is highly unstable and often holds more complex event cannot be defined by single junctions. I'm going to show an example for each chromoplexy and chromotripsis, but I just want to remind you this one slide that I showed before. And I said that with long-read sequencing, we can distinguish all these 3 different complex events. And I'm going to show you the evidence that with Severus, we indeed can do it. So this is a Severus output. It's an interactive graph. In here, each of these green segments are genomic segments, and each of these lines are junctions. And in this example, we have 31 kb insertions of chromosome 22 inside the chromosome 1 and inverted orientation. And this example is a foldback inversion from chromosome 14, comes and goes, and then goes to chromosome 5. So we have a small insertion and then it connects to chromosome X. This is a more complex event, a chromoplexy in ACC 1395 between 5 different chromosomes, leading to 4 different rearranged chromosomes in here. With Severus, we can also identify more complex events like chromotripsis, which is a reshuffling of an entire chromosome arm. In this example, it's a chromosome 21 in ACC 1954. If you zoom in this region, you can see all these junctions and segments involving in this event. Before long-read sequencing, chromatopsis event was identified through fluctuations in copy number. So next, we check the copy number profile of this region. We run Vahan. It's a haplotype-specific copy number caller for long-read sequencing. And here is the output. And the red one is haplotype 1, and the blue one is haplotype 2. Here is coverage and the copy number profile. Between 5 MB to 45 MB in here, you can see the fluctuations in copy number in haplotype 1, while in haplotype 2, it is pretty much stable, which also validates our finding for chromatopsis, and also that shows that with long-read sequencing and phasing, we can identify copy number profile specific to each haplotype. So as conclusion, with long-read sequencing, we can detect structural variations with high recall and precision. Severus works not only with tumor normal, but also tumor only and multi-site tumors. I didn't have time to go through, but with Severus, we also can provide phased calls. And at the last step, we provide complex SV clusters. Both Severus and Vahan are publicly available on GitHub. All the results that I showed today are from our benchmarking set, so the cell lines. And one question that you may ask about how it works in real tumor samples, but I'm a computational person, so I'll leave it to an expert. Lisa, she will continue with the clinical applications of these structural variations. Finally, I would like to thank all these people, and especially Kolmogorov, Midhat, and Veer. So thank you for all, and I'll leave it to Lisa.
Well, thank you, Ayse and Midhat, for the introductions. What I am going to be covering for part 3 of this talk is how we're able to use Severus to detect structural variants within the pediatric leukemia cases. And so what I'll be highlighting is a pilot project that we have performed, and this is looking at 11 cases of pediatric leukemia. And so the way that it was structured is we're looking at 7 cases of precursor B-cell acute lymphoblastic leukemia, and then 4 AML, they're acute myeloid leukemias. Within this study, what we did is we selected 5 known cases. And so we're really focusing on translocation events through Severus, despite the fact that, as Ayse nicely shared, we can see multiple different copy number events, for example, as well as some of these translocation events. But we're really hoping to deep dive into those translocations. And so with these known cases, there were 2 BALLs that had known translocations that are clinically relevant. And then also in our AMLs, we had 3 AMLs with clinically relevant translocations pictured here. In addition, we wanted to explore some of our unknown cases. And so these are cases that, despite having routine cytogenetic and molecular genetic testing, we weren't able to perform or identify a primary genetic driver that was underpinning those leukemias. And so we wanted to see whether or not Severus might resolve some of those for us. So with this study, what we did is we performed paired tumor normal testing. And so that really is a strength of the pipeline, where we're able to show true somatic translocations with this approach. We did use existing DNA. So this would be using about 5 to 7 micrograms of DNA per sample. And these were isolated through a variety of different isolation techniques, both automated and manual. And since these were leukemic cases, these were performed primarily on fresh blood and marrow. Then we performed HiFi sequencing using the Revio, as Midhat kindly introduced you all to. And this was using a standard smart library prep and also using 1 flow cell per sample. So that results in about 30x depth of coverage, which is sufficient for our structural variant detection. Then we ran each of these through the PacBio Human WGS workflow, and this was by performing, in kind of broad strokes overview, haplotagged alignment and phasing. And finally, we finished up with the structural variant calling using Severus, and this is where we really focused our analyses to date. So the goal was to look at the break ends specifically, and these break ends are the ones that are signifying whether or not there's a translocation that might be present. And we went through any of the break ends that were called for these cases and manually assessed whether or not there were any cancer-associated rearrangements. What we are really excited to see is that we were able to resolve for all 5 of the known cases, we were able to detect the translocations that were present, which was very exciting for us. And so 5 out of 5 were detected. And what I'm going to do is walk you through one of them just to give you an idea of what these data look like and how we're able to detect them within the cases. And so here what you're seeing is this is going to be an IGV plot. Hopefully with some of the introduction that you received earlier in today's talk, these will be a little bit easier to look at. But on this left-hand side, what we're seeing is a region on chromosome 21, and I'll bring your attention to the bottom. So this is looking at the RUNX1 gene on 21q. And then here what we're seeing is chromosome 8, and at the bottom is going to be RUNX1T1. So if we look at this track here, what this is going to be showing is our Severus call. And so you can see that there is a call made by Severus designated by this blue bar in both of these screenshots. And then what we're able to do, if we would zoom in, and I'll show you in just a minute what I mean, that there's going to be some of these reads that are going to have some annotations on them within this IGV screenshot. And so what these are going to show is that you can see within RUNX1 gene on chromosome 21 that some portion of these long reads are going to map to chromosome 8. And then similarly, some portion of the chromosome 8 reads are also going to be mapping to chromosome 21. And what we're able to see when we look at these data in a little bit more detail and also through our Severus calls is that this is indicative of this RUNX1P1 fusion. So in addition, what this project was able to do for us is this case of AML with a NUP98 translocation. It had been identified by FISH, was considered to be diagnostically resolved in that NUP98 is one of the more common genes in these fusions. And then what we could do is we actually found the fusion partner without any additional follow-up just by doing this HiFi sequencing and using Severus. And so we identified that the fusion partner was NSD1. In addition, each of these, although they had been detected through a variety of more conventional testing methodologies, what we were able to do with our HiFi sequencing was to find the precise breakpoints as well as confirm that exact partner gene. So then the even more interesting part, now that we had proof of principle that this was able to be successful in the detection of these translocations in our hands, is we wanted to look closer at those 6 unknowns. And what we were really excited to find is that 2 of our 6 unknowns could be resolved using this approach. That was a little bit higher than we were even maybe anticipating. We know that there are translocations that are present at a relatively high frequency within pediatric B-ALL and AML cases. However, these truly were diagnostically unknown from a genetic standpoint. And so we were hoping to detect some, but weren't sure if any would shake out. So having 2 was amazing. With these 2, I'm going to walk you through them in a little bit more detail, but the reason why they're so important and why we're so excited to have found them is that they do assist in binning our patient's particular cancer within a WHO diagnostic category. And so what you're seeing here is that KMT2A rearranged AML is a subtype of genetically defined AML. And then similarly, the ZNF384 rearrangement, that's going to fall within this WHO subtype of B-ALL with these other defined genetic alterations. So the big question we had is, well, why were these not originally detected? And so that's what I'll be walking through on the next few slides, is looking at both of these cases in a little bit more detail. So looking at our KMT2A MLT10 fusion case, we did perform chromosome study, and as you can see here, we did observe some atypical findings that are suggestive of this being a neoplastic cell. And so here, I think even to the untrained eye, it's fairly clear that there's a little bit more happening on 7p than is expected. I think that seeing this rearrangement with 3q is a little bit more challenging for some people in the audience. And then this is the same metaphase spread that you're observing just in karyotype form. But what's interesting is that we aren't weren't interested in either of those genes in terms of KMT2A or MLT10, but for those of you a little bit less familiar with where these reside, we're really looking for abnormalities that would be present on 10p for MLT10 and 11q for KMT2A. And so we were not able to observe anything that was obvious, at least with regards to any sort of translocations here. That's not necessarily atypical for this type of rearrangement, where they have been shown to be cytogenetically cryptic historically. But what was a little bit atypical for us for this case is that we also fished for KMT2A. And so with this KMT2A FISH probe, what you're observing here is going to be an ideogram of chromosome 11 and the approximate location of these probes. Please note they are not to scale, but for the sake of this presentation, they're a little bit larger than they should be. And then here, what we are expecting to see is we have a 5' green probe that is going to be on a portion of KMT2A, and then the 3' probe is going to be red. And what we observe within this nucleus that is pictured here is that, yes, there's a little bit of separation between a 5' and 3' probe. However, this is not significant enough of a separation for us to consider this to be broken apart. We'd expect them to be much further apart within this nucleus. And then these are even closer, even more compelling to say that they are not really separated in a clinically meaningful way. And so by our eyes and in our hands, we had designated this as KMT2A negative and had done our due diligence in fishing for this case. So what we were able to see by our HiFi data is really interesting, because not only does it tell us what is rearranged, but it can start to tell us how as well. And so what you're seeing here is going to be chromosome 10 on the left, chromosome 11 on the right, and then hopefully a useful diagram, though we will be diving into this even a little bit more granularly on the next few slides, where we can zoom into this region on chromosome 10 where MLT10 resides. And so what we're seeing in this plot is that we have Severus calls that are going to be on either side, and these are indicating a structural variant event that is occurring here within this region. And if we look at the bottom portion of this screenshot, we can see MLT10 is going to be right here on the left-hand side of the screen. ARMC3 is the gene that's on the right-hand side of the screen, and this is going to be about a 1.3, almost 1.4 megabase segment. And so what these reads are telling us, if we would zoom in here, and I apologize that we're not doing that for the sake of today and time, but this entire section of about 1.3 megabases is actually inserted into 11q right within the KMT2A gene. And so what this looks like in more of a diagram form is that here we have our chromosome 11, again with that green probe and the red probe on that 5' and 3' portion of KMT2A respectively. Then if we would tip chromosome 11 over on its side so that we have the P-ter over to the left and the Q-ter to the right, and then zoom in on this region that is being probed for, what we can see is that normally there's going to be about 40 kb in between the two probes. However, when we have this insertion of sequence from MLT10 through ARMC3, that's actually going to expand the distance between these two probes to about 1.4 megabases if you calculate out kind of as close as you can to the exact base pairs that this is happening within. And so that 1.4 megabases, although it sounds kind of large from a molecular genetic space, in a cytogenetic space, and especially a FISH space, that's actually not terribly large, where we are expecting something more on the magnitude of a 3 megabase or greater would be where we could see true separation, though this does vary a bit depending on your probe design. And so we did go back and look at the images that were captured as well as FISH for this case, and we really weren't able to observe any sort of separation that looked like it would be clinically significant, at least not in our hands. So then the final question that we had is, well, does this actually create a functional product? Because that's also really important when we're talking about these translocations. And what was really exciting for us from using this HiFi data is that we were able to determine exactly where these breakpoints were occurring. And so what we saw is that we could walk through the sequence data from the 5' end of KMT2A until we reached intron 8. Then we could see a break and then the insertion starting with intron 9 of MLT10 through the 3' end of MLT10, in addition to the kind of these additional intervening genes through ARMC3, and then it would pick right back up at intron 8 of KMT2A through the end of chromosome 11q. And so what this did is it confirmed for us that these were in the correct orientation to result in a functional product, which is what would be expected to actually be a driver mechanism for this patient's B-ALL. And so the other question that we had is, well, what would happen if we would fish for MLT10? Because we have a FISH probe that is a break apart probe that covers the 5' portion of MLT10 and the 3' portion, and only this 3' portion is expecting to be inserted into KMT2A. And so we did do follow-up testing to confirm our suspicions that this was real. And so what we observe here is the MLT10 probe with 5' in red and 3' in green. And we do see this very clear separation of these probes, indicating that it does look like MLT10 is rearranged. And then that also supports our HiFi findings, which is really exciting for this case. So in summary, why was this particular case not originally detected in terms of that KMT2A MLT10 rearrangement? So cytogenetically, from both a karyotype perspective and from the FISH testing that we performed, it was cytogenetically cryptic. Now, that said, had we fished for MLT10, we certainly would have had some suspicion about what was going on for this case. And so that's something that we're keeping in mind for additional cases that come back as negative for our ALL FISH panel. Then in terms of microarray, this is a balanced rearrangement, so we weren't able to detect it there. Our in-house cancer NGS testing is currently SNV only, and this is using exomes. So we did not detect structural variants by design. And then for RNA testing, unfortunately, RNA was unavailable for this case. So it really did sail through our routine workflow undetected until we were able to do this follow-up study. So then looking at our other ZNF384 TCF3 fusion case, again, we're looking at these IGV screenshots that should hopefully be a little bit more familiar now. On this left-hand side is chromosome 12 overlapping ZNF384. On the right-hand side is chromosome 19 overlapping TCF3. Again, we have our Severus calls, and hopefully you can appreciate when we're looking at this depth plot from IGV that there's actually a drop in coverage here and a drop in coverage here, indicating that there's likely some sort of structural event that is occurring. And then what you're also seeing is this is what we are looking at in terms of phased reads. And so we're trying to split apart the reads that seem to correspond with a translocation versus those that do not. And so here we can see support for chromosome 19 material being present within chromosome 12. And here we can see support for chromosome 12 material being present within chromosome 19 and really indicating that we have a fusion between these two genes. So this case, why was it not detected? Well, on karyotype, this particular translocation is cryptic, so we're not expecting to be able to pick it up. On our in-house FISH panel, we don't test for either of these genes. That's because it's a relatively rare translocation, though we could always consider adding in a probe eventually down the road, depending on how many more of these we pick up, for example. On microarray, it is balanced again, so we're not expecting to detect it on microarray. And then on our exomes in-house, it would be an SNV only analysis, so weren't able to detect structural variants there. And then what was interesting for this one is we said, wait, we did perform RNA-based fusion testing. Why was it not detected? And when we looked at the genes that were included on the fusion panel, it turns out that neither of these genes were included. So again, sailed through without being flagged at all by the technologies that we employed, again, in our hands, and then with some of the send-out testing as well. So in summary, what we found is that we were able to identify known translocations in 5 out of 5 cases. And then for 2 of our 6, we were able to come up with primary diagnostic categories for 1 AML and then 1 B-ALL case. So I did want to take just a few more minutes to talk about our future directions. And so one note is that we're hoping to examine other genetic variant types. So these would be things like inversions, deletions, duplications, all of which can be detected and called by Severus. We're especially interested in doing so for the remaining 4 unknown cases. And so in one of those 4 unknown cases, what we have already seen is that it does hold quite a bit of promise. Here we're seeing Severus calling in amplification that's overlapping PAX5, for example. So we really are hopeful that using this approach will help us resolve a few more of those unknowns. In addition, we're really excited to dig into the data even more and identify other types of somatic variants. So we are just starting to scratch the surface with these data, and we still have sequence variants, methylation changes, mutational signatures that we could analyze. And so this is a little bit busy, but I think it's very informative. Then it really outlines all of the different ways that these data can be processed and analyzed. And so we start with our HiFi reads, and after they're aligned, and again, this is a tumor normal workflow, so we're really looking at true somatic variants. We can do structural variant calling, we can look at phasing, we can do methylation, we can do SNVs, and not wanting to really spend too much more time here, but just wanted to show that this is a very powerful approach that if performed in either a paired tumor normal way or even potentially in a tumor only way, could be really useful in the clinic. Then what we're hoping to do is evaluate our tumor-only analyses. So we realize that it's not always possible to get a paired germline sample. And in those cases, it's very useful to still perform tumor-only and sort of see what you get, what you can parse out. And so one of the powers of this HiFi sequencing or long-read sequencing in general is that you can start to tease apart what would be called as sort of a mosaic structural variant and determine whether or not it's happening at a clonal versus subclonal VAF, how that variant allele frequency or VAF correlates with your percent tumor that you are sequencing. Note, we did make sure that we had high percent tumors for all of the cases in our pilot study. There were on average around 80% tumor, some upwards of 90% tumor. So that's something else that we're actively investigating, is what's sort of the lowest percent tumor that could be used where we can still successfully detect these structural variants, for example, with a single flow cell at that 30x step. The other thing to note is that by doing tumor only, over here what we're looking at is the number of break-ins that are called by Severus, and tumor only really does significantly increase the amount of noise to sift through for a particular analysis, whereas when we use the match normal, we're able to very quickly whittle down the list of true somatic calls. And that said, we're confident that this number can be relatively easily decreased by employing, say, gene panels or looking only for specific breakpoints, for example, that are clinically meaningful. And then finally, what we're really hoping to do is expand this to other tumor types. We've started these sequencing efforts, have the data in-house, but are looking for the time with which to analyze them. So please stay tuned, and then we'd like to evaluate additional cases. And then for some final parting thoughts, we also realize that it is a balance when looking at what assays to employ, both clinically and on a research basis. It's this balance between time versus money spent, especially when talking about pediatric cancer or cancer testing in general. And so there is this battery of tests that is currently available and widely used in multiple clinical labs, all of which are very valuable in tracking down diagnostic findings, many of which are pictured over on the right in pediatric leukemia. However, what we are really interested in exploring further is whether or not we might be able to reduce or collapse some of this testing down into a single test, while keeping in mind that there's this additional value of, say, the single cell nature of karyotyping and FISH technologies that to date is not going to be easily replaced by this long-read sequencing. So really excited to dig into that further and see if we can come up with a test algorithm that works well for our lab. And we know that it will vary lab to lab as well in finding the right fit. So with that, we'd like to acknowledge a number of different individuals who have made all this work possible, especially from Children's Mercy, PacBio, also from the NCI, and then really a huge thank you to patients and families who are willing to enroll in these research studies, as well as a variety of different funding sources that have made this project and all of this work possible. And with that, we'd be happy to take any questions. Thank you.
Well, thank you to all the speakers. I also want to thank Dr. Farooqi and Dr. Keskus. They're answering questions in the Q&A. You guys have asked a lot of great questions. So please refer to the Q&A because there's some really great answers with nice length of discussion for any of the questions. I think we have time for maybe one question, unfortunately, so that we can give you the codes for for getting CME. So the question that keeps coming up a little bit is, what is the limit of detection with 30x coverage, and how does this give you a sense of the actual variant allele fraction?
Yep. So I answered this one in the chat, but I am estimating limit of detection to be 20% tumor, 10% variant allele frequency at 30x coverage. This is again done sort of tumor normal together and HiFi. I feel we can pretty comfortably call a break end with 2 reads in the setting. And so 20% tumor, 10% VAF, 30x coverage, we would in theory get 3 reads and so cross that threshold. But as Lisa mentioned, we have to do the formal, we plan on doing sort of former tumor normal dilution studies. And maybe she or Ayse can talk a little bit more about this. We're also planning on like in silico dilution studies to test Severus.
Awesome. Thank you so much. They're going to continue to answer some questions in the Q&A, but I just wanted to thank everyone for attending as we hit the 10 o'clock, the hour. The webinar code for the PACE credit is listed here. Please, complete the session evaluation by May 3rd to get the credit. And again, some reminders, please attend the CGC 2024 annual meeting. Early registration ends on May 21st. And again, May 21st is a big date because there's another webinar that we'll be hosting. The CGC Education Committee is very proud to host all these webinars, and we're very lucky to have this webinar today because it was a great, excellent webinar. And then finally, please encourage your fellows to submit interesting cases. It gives them national recognition, giving talks in front of, essentially an international crowd. And it's a CME granting. So it's another thing to put on their CVs. So please scan this QR code, send it to your fellows, and we'd be very thankful for that. And with this, I again want to thank the speakers for an excellent discussion. There were so many people that joined, and I'm just so happy to offer these things from the education group at the CGC. So thank you, speakers, and thank you to everyone. Have a wonderful day.